you so much, Sayun, for inviting me to give this plenary. It's a real pleasure and a tremendous honor to be with you to spend the next 45 minutes or so to tell you about some of our work, some of the work going on in my laboratory. I always love coming to Korea. It's one of my favorite places to visit, and I've already enjoyed my very brief visit. So thank you all for joining me. I'd like to make this as informal as possible, so please ask questions, just like this morning, in English. Interrupt whenever you need. What I'm going to try to do in the next 45, 50 minutes or so is cover an introduction to make sure we're all on the same page about some of the basics, and then tell you 3 stories on drug development, on discovery science, and on developing combination therapies for cancer. And I don't have time to show you all of the data, just going to give you a vignette, a summary. I'm happy to go over details later on if anybody's interested. So, just to cover some of the background and the basics, this is the pathway that I have been preoccupied with for, over 30 years now, since I was a postdoc, which is the PI3 kinase AKT signaling pathway that operates in all cells and tissues in normal physiological signaling. As most of you, I suspect, know, this pathway responds, as all pathways do, to extracellular stimuli, whether they be growth factors or hormones or neurotransmitters, ligands that engage receptors at the surface, which through a complex set of mechanisms culminate in the activation of a family of enzymes collectively known as the PI3 kinases, so called because they interconvert a minor membrane phosphorinositide, a lipid, PI45P2, into a second messenger known as phosphatidylinositol 3,4,5 trisphosphate. This is the most negatively charged lipid in the cell. And that immediately tells you something about its function. This is generally not a structural lipid. It is not required for the integrity of membranes. It is a signaling molecule. And it fulfills all of the requirements of a second messenger transducing molecule. It is absent in unstimulated cells. It rapidly accumulates at sites of synthesis where PI3 kinases are activated. And it is equally rapidly degraded or removed by active enzymatic mechanisms, which in this case is an enzyme that catalyzes the reverse reaction of PI3 kinase, an enzyme known as PTEN, which is one of the most frequently mutated tumor suppressors in cancer. Once PIP3 is synthesized at the membrane, it then recruits a whole host of downstream effectors, which are normally in the cytoplasm, recruits them to the membrane. They then undergoes a conformational change. And one of the most studied effectors, easily the most studied effector in this pathway, is a serine-threonine protein kinase known as AKT. Once PIP3 binds to AKT through a very well-understood lipid-protein interaction, it undergoes a very remarkable conformational change, which exposes the catalytic kinase domain. It is phosphorylated by complex mechanisms that we also understand with exquisite knowledge. The enzyme is now locked in the catalytically competent conformation and can fall off membranes and translocate to multiple intracellular locations, including the nucleus, Golgi, mitochondria, mostly the cytoplasm, where it catalyzes its function. And its function as a protein kinase is to phosphorylate any one of about 200 or more substrates which catalyze downstream signal relay. Now, for the first 25 or so years in this field, we largely assumed, as it turns out incorrectly, that all of this activity occurs at the plasma membrane, which is where PI3 kinases are localized, where all of the enzymes required are localized. We now know through the work of many laboratories that in fact this is wrong. There's a lot of activity going on at endomembranes, particularly in the endosomal compartments, where PI3 kinases are localized, where PIP3 is made, where AKT is recruited, where a lot of the regulators of this pathway are also localized. AKT is activated and transduces the signal. And you can imagine that the signals that emanate from endomembrane signaling compartments are going to be very different from those that emanate from the plasma membrane. We understand what goes on here in a lot of detail. We understand what goes on here in a lot less detail. AKT is regulated by very complex mechanisms, not just phosphorylation at two key residues in the catalytic kinase core, but a whole host of other post-translational modifications, including additional serine-threonine phosphorylation, tyrosine phosphorylation, even weird and wonderful modifications such as lysine acetylation, ubiquitylation, sumylation, even sugar modifications. We really don't understand what many of these modifications do to the activation cycle of AKT, but they have been documented. There exist 3 AKT isoforms in mammals. They are distinct gene products that reside on different chromosomes, but with a very similar modular structure, as depicted here, and a very high degree of identity, which for the first 30 years or so of work led most people, including myself, to believe that these had largely redundant functions in both normal cellular physiology and in disease. But as was first recognized by Maury Birnbaum, who made the very first AKT1, AKT2, and AKT3 whole animal knockout mice that showed very distinct and non-overlapping phenotypes, we now know that AKT1, 2, and 3 have non-redundant, non-overlapping phenotypes in both normal physiology and in disease. For example, AKT2 knockout mice develop an insulin-resistant and diabetes-like phenotype, which you don't see with the AKT1 or AKT3 knockout mice. They have other distinct abnormalities, although all are viable, developmentally viable. And through the work of my lab and many other labs, we also know that AKT1, 2, and 3 have very distinct activities in pathophysiology, such as cancer, where AKT1 and 2, for example, have distinct roles in modulating cell migration, cell invasion, and metastatic dissemination. We understand these quite well. That's all I'm going to say about AKT1 and 2 and 3 non-redundant functions. Now, going back to the wiring of the signaling pathway, as illustrated in this diagram that I've taken from a review that I wrote with my friend Brendan Manning about 3 or 4 years ago, covering 25 years of research in the field, once activated by this complex mechanism, AKT transduces a signal to a large number of substrates. But as we argued in this review, AKT transduces a signal to what we argue are 3 evolutionarily conserved multifunctional nodes comprising 3 very important substrates, 3 very important gene products: the fork-head BOXO or FOXO family of transcription factors, the glycogen synthase kinase 3 enzyme, and the TSC2 protein of the tuberosclerosis complex. All 3 are multi-site phosphorylated by AKT, and somewhat counterintuitively, at least at the time, phosphorylation by AKT serves to inactivate their function. Now, you would think for an oncogenic signaling pathway, phosphorylation would serve to activate, but in fact, for many AKT substrates, their phosphorylation serves to inactivate their function. For example, phosphorylation of FOXO by AKT serves to retain it in the cytoplasm, excluded from the nucleus, thereby terminating transcriptional activity. For GSK3 phosphorylation, this is a constitutively active kinase, and phosphorylation by AKT serves to inactivate its catalytic activity. And the same is essentially true for the TSC2 protein. Now, there are, of course, or as I said earlier, over 200 substrates of AKT in a given cell or tissue, and a few of them are depicted here. And although many are inactivated by phosphorylation, this is not the rule. There are many whose phosphorylation leads to their activation, so it's an on signal as opposed to an off signal. Ultimately, collectively, all these proteins regulate every single cellular phenotype that you care to imagine, both cellular survival, cellular proliferation, as in cell division, metabolism, cell growth, as in cell size, and many other phenotypes, all of which are deregulated in human pathophysiologies, including cancer. And the reason this pathway has gained so much attraction from the basic science community, from the drug development community is because it is the most frequently deregulated pathway in all of cancer. In aggregate, if you query TCGA datasets for a number of the key rate-limiting gene products that participate in PI3 kinase AKT signal relay, including PI3 kinase itself, AKT genes, some of the substrates, the tumor suppressor PTEN, in aggregate, and you query for mutations, somatic mutations, gene fusions, gene amplification, copy number gains, or deep deletions in tumor suppressors, you find that in aggregate, this is the most frequently genetically deregulated pathway in all of cancer. Okay? To the extent that the two genes that participate in PI3 kinase signal relay, PIK3CA, which is the P110 alpha subunit of the class 1A PI3 kinase, which is an oncogene, is the number 2 most frequently mutated oncogene in all of cancers, second only to KRAS. Whereas PTEN is the second most frequently inactivated tumor suppressor in all of cancers, second only to P53. And this is especially true in women's cancers. So endometrial cancer, uterine cancer, breast cancer, ovarian cancer, cervical cancer are littered with genetic aberrations, genetic mutations in genes that participate, whose protein products participate in PI3 kinase signal relay. For this reason, the pharmaceutical industry for the last 25 years or so has been very hard at work to develop small molecule inhibitors to interrupt this pathway for therapeutic benefit, with amazing success, at least at the drug development level, to the extent that there are multiple PI3 kinase isoforms, so pan-PI3 kinase inhibitors that inhibit multiple PI3 kinase isoforms have been developed. Isoform-selective PI3 kinase inhibitors have been developed. There are, of course, dual PI3 kinase mTOR inhibitors, as well as catalytic and allosteric mTORC1 inhibitors, sort of rapamycin and rapalogs, and AKT inhibitors, which is where I'm going to turn my attention to for the rest of the talk. Unfortunately, the clinical outcome of the billions of dollars that have been thrown at these drugs have been remarkably disappointing, to the extent that the only 2, actually 3 PI3 kinase inhibitors have been approved by regulatory agencies. The first one being idelalisib, also known as CAL101, which is a P110 delta inhibitor, which was approved for the treatment of chronic lymphocytic leukemia, so hematological malignancies. And many of the other inhibitors have failed, at least as monotherapies in clinical trials, for a number of reasons, most prominently dose-limiting toxicities. And in fact, there was a remarkable excitement in 2019 when Fabrice André and his team had the very first a phase 3, the SOLAR-1 clinical trial of a PI3 kinase alpha selective, or PIK3CA selective inhibitor, alpelisib, which showed a doubling of progression-free survival in this phase 3 trial, combined with the endocrine therapy fulvestrant in ER-positive, estrogen receptor-positive breast cancer patients with a PIK3CA oncogenic mutation, compared to patients treated with placebo plus endocrine therapy. So a doubling of progression-free survival, okay? And this led to the approval of this combination in postmenopausal women with ER-positive disease, oncogenic PIK3CA. I just want to point out for the many trainees in the audience, this was in 2019. The discovery of the enzyme, or the enzymatic activity, I should say, that this drug targets, which is PI3 kinase, that discovery was 40 years before this. This in the mid-1980s when Luke Hantley and colleagues identified a PI3 kinase activity in fibroblasts. It took 40 years of research, drug discovery, clinical trial, and drug development to lead to this approval. So that's a sobering concept, and this is not an unusual timeline for drug development. I should point out that this combination, whilst showing promise, is only third-line treatment. In the real world, patients do not tolerate this combination very well. This is post-chemotherapy. This is post-CDK4/6 treatment in the ER-positive setting, okay? So this is by no means a home run, but it is an approval of a PI3 kinase drug that people have been working on for 40 years. I will turn my attention now to AKT inhibitors. Why? Because AKT inhibitors to date have not yet been approved. Yet. A whole number of drugs have been developed that are exquisitely potent and exquisitely specific. These are some of the drugs that are under development. So the two lead compounds in the clinic are ipatasertib, a Genentech compound, and capivasertib, an AstraZeneca compound. These are catalytic ATP-competitive inhibitors with remarkable potency and selectivity, even for ATP-competitive inhibitors. There's a whole bunch of other ones that are also in clinical development, but probably will not make it. And there was real excitement about 3 or 4 years ago when the Lotus phase 2 clinical trial, of the Genentech compound ipatasertib in triple-negative breast cancer patients, this is a phase 2 trial, showed over a doubling, more than a doubling of progression-free survival in patients treated with ipatasertib plus standard of care in TNBC, paclitaxel, compared to patients treated with placebo plus paclitaxel. So this immediately led many people, including myself, to believe that this would be the very first AKT inhibitor to be approved by regulatory agencies. The Lotus phase 2 clinical trial immediately spurred the extension into the phase 3 trial, the Opportunity trial, same patient population, TNBC, same drug combination, ipatasertib plus paclitaxel, which showed absolutely no benefit whatsoever compared to placebo plus ipatasertib plus paclitaxel. And so this led Genentech, unfortunately, to drop this trial, this combination, this drug in the breast cancer setting. It is being actively pursued in other indications. And the question is why? Why did you have in the same patient population such a remarkable improvement in progression-free survival and no benefit in the phase 3 trial? We can argue about that, but that was the real-world experience. So why have these drugs why have they fared so poorly, either as monotherapy or as combination therapy? Well, we used to think that it's lack of selectivity. These are ATP-competitive inhibitors. All kinases have an ATP binding site. They all look the same. Poor selectivity would give you dose-limiting toxicity. That is not the answer. These are remarkably selective drugs. The real reason is toxicity, dose-limiting on-target toxicity. Hyperglycemia, hyperinsulinemia, and various other DLTs are experienced by most patients on these drugs. There are, of course, multiple nodes of feedback activation that account for dose-limiting toxicities in this pathway. And there are kinase-independent functions that have been ascribed to AKT that we really don't understand very well, and where an ATP-competitive inhibitor is going to be ineffective, making the assumption that maybe kinase-independent functions are relevant for pathophysiology, and we don't really know much about that. So armed with this information, the first story I'm going to tell you is, can we develop alternative strategies to drug therapeutically the AKT pathway using a different strategy? And this work, I'm going to tell you, has been a collaboration with my friend and colleague, Nathaniel Gray, who used to be at the Dana-Farber in Boston. He's now at Stanford. And one day, because our kids, they used to go to the same school, and so I'd hang out with him at the playground, and I used to talk to him about, he's a chemical biologist. In my view, the best chemical biologist in the world. And we would talk about making drugs. And I said to him one day, and he got into the degrader business, in the PROTAC business. And I said to him, has anyone made an AKT degrader? He goes, no. And I said, well, could we make one? He goes, okay. So he went back to his lab and he spoke to his grad student, Inchel, and I went back to my lab and I spoke to my grad student, Emily. We got together and made the very first in-class AKT degrader. So what are degraders? Most of you probably know, but it's a very cool pharmacology where you have two, these are strange drugs. But there's a very clever technology that was first devised, iterated by Craig Crews and his colleagues at Yale, where you have two warheads, they're called. One is a ligand that engages the E3 ubiquitination machinery. The other is a ligand that engages your protein of interest. Okay? And then they're hooked up with a linker. Okay? And the idea is you throw these onto cells, and the degrader will bring your protein of interest, in this case AKT, in proximity to the E3 that the ligand will engage. And of course, this will then target your protein of interest to the proteasome, because it's an E3 ligase. It will ubiquitinate your protein of interest. The proteasome will degrade it. And now, instead of having catalytic inhibition, what you have is wholesale protein degradation. That's why these are called degraders, or proteolysis targeting chimeras. And so through some very clever medicinal chemistry and chemical biology, we made the very first in-class AKT degrader, and we published this a few years ago. I'm not going to tell you about it because what I'm going to show you is our second in-class AKT degrader. We published this in Cell Chemical Biology. If you want to scan this, it'll take you to the PDF. It was very potent and selective. It degraded AKT. But we noticed both in cells and in vivo, it had relatively slow kinetics. It took quite a long time to get profound quantitative AKT degradation. So the chemical biologists went back to the hood and devised the second degrader, which we hoped would have better improved kinetics. And I should say that for our first in-class degrader, we used ipatasertib, GDC0068, as the ligand to engage AKT, and we used cereblon as the E3 ligase. Actually, we used an IMID, lenalidomide, to engage the E3 ligase cereblon, which is a very common strategy. And so we made a second generation degrader, which also engages using the AKT inhibitor ipatasertib, but this time we used a ligand that engages VHL, the von Hippel-Lindau E3 ubiquitin ligase. And these are all unpublished, although this paper is technically in press and will be coming out hopefully soon in Science Signaling. And here is our second generation, and here's our first generation inhibitor. You can see that you put it onto cells in a dose-dependent manner. You get degradation of AKT at nanomolar concentrations. And you can see that it has improved degradation activity towards the first generation degrader, and it is much faster. So these are the kinetics. Within 4 hours, we see 50% degradation of AKT, but really the first generation degrader was much, much slower and nowhere near as quantitatively robust.
I'm wondering how you identify the inhibitor for AKT.
Why did we pick a particular inhibitor?
Yeah.
So I'm showing you what worked. What I'm not showing you is the fact that Inchel made about 50 or 60 different molecules, none of which worked, and they all had to be tested. We tried different drugs, we tried different linkers, we tried different rigidities. So it's an iterative process. A lot of it is guesswork. So what I'm showing you is what worked. There was no particular reason of choosing GDC0068 other than the fact that at the time, this was the lead clinical compound, and we knew it would work. That's it. And so you get this level of degradation in a both dose and time-dependent manner. It's exquisitely potent and specific. So this is a kinome scan where you measure binding of a drug to every single kinase in the human kinome. And you can see that the degrader actually only binds to AKT with a few small, relatively weak off targets, and it's actually improved over the parent catalytic inhibitor. This is not an uncommon finding with degraders with respect to their parent catalytic inhibitors. And to illustrate this even more, this is a proteomic scan where about 10,000 different proteins are scanned by quantitative mass spec. And you can see that with a twofold cutoff, the only 3 proteins that are degraded are AKT1, 2, and 3. These 2 additional ones are known to be AKT target proteins with a short half-life, so you would expect them to fall out. Everything else in the cell is essentially untouched. So exquisitely specific. And I know this is a busy slide. These are proliferation assays, okay? But the only thing that you really need to pay attention to are the 2 curves where you have the black line here and the orange line, which is essentially superimposed. These are the first and second generation degrader. They outperform, in terms of cellular proliferation, of a ER-positive breast cancer cell with an oncogenic PI3 kinase mutation. They outperform every single other catalytic or allosteric inhibitor of AKT under clinical development, all right? Sometimes by a log-fold or more. So these are the most potent AKT drugs, because they're not inhibitors, they are degraders, that we have yet tested compared to anything that is in the clinical pipeline. We then hooked up with AstraZeneca, who expressed an interest in developing this in a medicinal chemical pipeline. This is their Horizon breast cancer. This is their Horizon cell line profiling, which profiles thousands of cell lines across over 30 different lineages, including normal cells. And I know it's quite complicated, but it's color-coded. So the blue, for example, are breast cancer cell lines, okay? Yellow are endometrial. And the only thing you really need to know is that anything under the black line means that these cell lineages, these cell lines, are more sensitive to degrader than they are to the parent inhibitor. Okay? And in fact, whereas these are largely insensitive, in fact, it is no surprise that all of the cell lines here, when you look at them, these are all cell lines that harbor a mutation in a gene that participates in PI3 kinase signal relay, either oncogenic PIK3CA, PTEN inactivation, AKT amplification, etc. So what we've done now, and I don't have too much time to go into this, we have run our degrader against inhibitor using steady-state metabolomics, okay? Because one of the major mechanisms by which PI3 kinase AKT impacts tumorigenesis is by metabolic reprogramming, okay? And so we've done steady-state metabolomics using our core, and we have done RNA-seq, and this has taken a lot of optimizing, which is sort of shown here, and I don't have time to go into that. We treat cells over time with degrader and inhibitor or vehicle control, RNA-seq, and then what we did was use an algorithm known as COSMOS, which stands for Causal-Oriented Search of Multi-Omic Space, which is essentially a computational pipeline where you input data sets from RNA-seq and from metabolomics, and it runs on a cluster, on a supercomputer cluster, iterative processes which measures, and so this is the RNA-seq data, this is the metabolomics data, and it takes these data sets and runs them against the prior knowledge network of signaling protein metabolite interactions and vice versa. And you run this iteratively over many cycles, and it's a predictive algorithm. And so what you get from these analyses are these network maps which measure the interconnectedness between metabolites and proteins, metabolites, and proteins in a predictive manner based on prior knowledge. Okay. And so you get these networks, and so this is the network that came out with the AKT inhibitor, and this is the network that came out with the degrader. And what you're looking for are the most interconnected nodes, because that is different between inhibition and degradation of AKT. Okay? Because there has to be a reason as to why a degrader functions more potently and better than an inhibitor, which after all, you're targeting the same enzyme, the same kinase. So why is that? And unexpectedly, at least to me, the two most interconnected nodes that showed up in degrader versus inhibitor are MAPK14 and MAPK8. And these encode the stress kinases JNK and P38 respectively. And so now the experiments become somewhat obvious. You can run signaling experiments where you compare degradation versus inhibition, and you can see that sure enough, in cells treated with degrader, there is a potent upregulation of c-Jun, both total and phospho, which you don't see with inhibitor, and also with P38. Essentially, remember, the Cosmos analysis is only predictive. You have to prove it biologically, and that's what this does. And you can now take cells and treat them with degrader or inhibitor, which induces cell death, and you can co-treat them with a JNK inhibitor or a P38 alpha inhibitor, and with a JNK inhibitor, you can now rescue degrader-induced cell death, whereas a P38 inhibitor does not. So this argues that there is a reason that AKT degradation leads to induction of JNK, causing cell death, and you can rescue this with a JNK inhibitor. The question then becomes why? Why does degradation of AKT, but not inhibition of AKT, lead to activation of JNK? And we really have no idea. You could argue scaffolding, you could argue you can make all sorts of hypotheses, but this is largely a hypothesis-generating engine. These kinds of analyses are hypothesis-generating engines.
So can you correct for, I guess you can't do a standard kind of inhibitor to inhibitor comparison in vitro because one of them is degrader. But if you look at AKT inhibition using AKT phosphosubstrate as a readout.
So that's a great question. So the question is, how do you normalize for inhibition versus degradation? The whole point is that when we did our we took a standard readout for AKT, in this case Pras40, and we ensured that we had equivalent inhibition of Pras40 phosphorylation with degrader versus inhibitor, as best as we could approximate. So that the reason you get differences is not because you have differences in potency, but because it's something beyond potency, such as a biological event, if that's what you were asking.
The cells are always balancing between survival and express. Maybe if you shut down the cell signaling for cell survival, then the stress signaling goes up. So maybe because of that.
That could certainly be the reason. The question is why? Why in response to degradation do you see this and not with inhibition? Is it just a potency thing because it's a better drug? We don't think so. But that's to be determined. Yes?
Is the GDC inhibitor reversible or irreversible?
The GDC inhibitor is reversible. It's not a covalent inhibitor. There's only one covalent inhibitor, berusaterbib, and we haven't tested that.
Do you think this change, this difference between the reversible and degrader can make a change from that?
So I don't have time to show you, but you can go to the paper and look at it. We've done washout experiments, so we can wash out the drug, at least from cells, and you maintain degradation. In fact, and again, I don't have time to show you this, the remarkable thing is that, I don't have the slide, you can put degrader onto cells at time 0. You see degradation within 2 hours. With a single dose, you can come back after 2 days, after 3 days, after a week, and you still see degradation. Now that cannot be because there is still drug. The drug is metabolized within a couple of hours. We know that. So there has to be some feedback mechanism which accounts for the continuous degradation or perhaps lack of synthesis of AKT, which is why this is such a potent drug and such a long-lasting drug. Move on to the second part. This is all published, but Seyoon wanted me to present this, so I'm going to present it because it's one of the things that I'm perhaps most proud of in my career. And that is a paper that we published about a year ago now, which deals with the reprogramming of cancer metabolism by the PI3 kinase pathway. And the pathway that I'm going to focus on is coenzyme A. Okay? Coenzyme A is the major, I would argue that coenzyme A is probably the most important cofactor in all cells and tissues. There are others. And it's a universally conserved mechanism as a carbon shuttle. All right, it shuttles carbon. It is functional in obviously the mitochondria for the TCA cycle, at least when coupled to acetyl groups as acetyl-CoA. It is required for fatty acid biosynthesis, beta oxidation, and perhaps what you're most used to is the modification of proteins by acetyl groups. For example, histone acetylation. The entire chromatin code is driven by the post-translational modification of histones by acetyl groups. All of this derived from CoA and acetyl-CoA, which is fueled by glycolysis pyruvate to generate acetyl-CoA. And so this project began many years ago by a remarkably talented postdoc in my lab, Christian Dibble, when he did a very simple metabolomics experiment, steady-state metabolomics experiment, taking standard epithelial cells, treating them with insulin, which activates PI3 kinase, pre-treating them with a potent PI3 kinase inhibitor, and then doing steady-state metabolomics. And what you get are metabolites that change according to what has been published by many labs over the last 10, 15 years. That is to say, glycolytic intermediates in the pentose phosphate pathway, many intermediates in nucleotide metabolism. But there was one metabolite that we didn't even recognize by name, and that is pantothenate that accumulated in cells treated with PI3 kinase inhibitor. So what is pantothenate? It's also known as vitamin B5. And as Dr. Kim presented this morning, this is one of those curious and unusual pathways. It's very linear and simple, relatively simple as metabolic pathways go. Begins by cells taking up vitamin B5, pantothenate, and there is the input of cysteine, ATP, the AP moiety of ATP, to generate what this is, which is coenzyme A, cysteine moiety, pantothenate, and ATP. With a very ordered series of events with enzymes that we know very well, at least in most cases. What is pantothenate? Well, it's, as I said, also known as vitamin B5. We have to take it up in the diet as mammals. We cannot biosynthesize it ourselves. These are some of the foods that you eat every day, maybe, and contain vitamin B5, or you can buy it at your favorite health food store. We need about 5 milligrams per day for adults. And as far as we know, in fact, this is pretty certain, the only use of vitamin B5 is to make coenzyme A. That's it. That's all it is used for. And it actually comes from the Greek pantos, which means from everywhere, because it's almost in all foods that we eat every day. Okay, so what I'm going to show you now very, very quickly, because this is all published, and I want to make sure I present you the final story. This is all published, a series of data that show you how insulin stimulates PI3 kinase AKT to produce coenzyme A, and mechanistically how this works, and what it means for cells, especially cancer cells. So instead, these are, for the aficionados, metabolomics, flux analyses, where we feed cells with labeled, carbon, nitrogen labeled vitamin B5, and then we trace the carbon and the nitrogen through the biosynthetic pathway all the way to CoA and acetyl-CoA. Okay? So you stimulate cells with insulin, you make CoA and acetyl-CoA. You treat cells with structurally unrelated PI3 kinase inhibitors, you completely block this de novo biosynthesis. You take cells where we've engineered them with an oncogenic AKT mutation that is hyperactive. Okay? In the absence of insulin, these cells constitutively make CoA and acetyl-CoA. You treat, and this is in a doxycycline-inducible manner, you treat them with a catalytic AKT inhibitor, you completely block this constitutive production of CoA and acetyl-CoA, and rapamycin generally has no effect, arguing that this is downstream of AKT, but not as mTORC S6 kinase signaling. Mechanistically, how does this work? So for this, you need to know the biosynthetic route and the enzymes involved. There are 4 enzymes, the pantothenate kinases and these 3 other enzymes. And if you scan the protein sequences of these enzymes, and if you scan phosphoproteomic analyses, you find that PANK1, and PANK4 in particular have high fidelity AKT consensus motifs that will be predicted to be phosphorylated by AKT, that are not found in any of the other enzymes that are required for coenzyme A biosynthesis. So the experiments then become very simple. You can show that PANK2 and PANK4, but not PANK1, are phosphorylated, as shown here, in response to insulin, and here, and this is blocked by a PI3 kinase inhibitor. We don't know if PANK1 is phosphorylated. It could simply be, even though it has a very good site, it could simply be that this antibody does not recognize this site. And so functionally, we started asking questions. There are 4 pantothenate kinases in cells: PANK1, PANK2, and PANK4. PANK3 is mostly catalytic and is not expressed in most cells, so we forgot about that. And we simply did an experiment where we treated cells with siRNA to PANK1, PANK2, and PANK4, and then measured CoA, acetyl-CoA, in every single CoA species in cells by steady-state metabolomics. And if this was a forward mechanism, a positive signal, you would expect that knocking out PANK1, the pantothenate kinases, which interconvert pantothenate vitamin B5 to phosphopantothenate, you would see a reduction, and sure enough, that's what we see. You see a reduction of all CoA species. But when you knock out PANK4 in the same cell, same exact experiment, you accumulate CoA and CoA species. This is very paradoxical. You would not expect this. This is indicative of a suppressive mechanism for the endogenous protein. And how could you explain this? Well, and this is not only at the level of metabolite, but also at the level of cell growth. It's a complicated experiment. We've taken MCF10A epithelial cells, which have a knock-in mutation in AKT locus at E17K, which is a mutation found in patients. This is hyperactivating AKT. We knock out, using CRISPR-Cas9, PANK4, and now we can put back PANK4, either wild type or mutants at the phosphorylation sites, and you can see that if in the vector cells, these are normal knockouts of PANK4, you put in the wild type, you actually suppress proliferation. So this is arguing that this is some sort of a tumor suppressor, or at least a growth suppressor. Okay? And the reason this makes sense, because this didn't make any sense at all. If it's a kinase, it should be giving a positive signal. Turns out the PANK4 is not a kinase. It's a pseudokinase. It lacks the residues required for catalytic activity. Instead, what it has is a phosphatase domain, a DUF89-like phosphatase domain with aspartic acid residues that would predict it to be catalytically active. And if it's now a phosphatase, you can immediately think and see why this would be a suppressor of normal cell growth. And now the experiments become obvious, because you can make the mutations in the aspartic acid residues, and when you put in wild-type PANK4, which suppresses CoA and acetyl-CoA, you can now completely inactivate this activity or rescue this by making the phosphatase catalytic mutants for both total and M+4. And so now a series of experiments that prove that the phosphatase activity of PANK4 is required here. Again, the wild-type PANK4 suppresses growth, the phosphatase mutants rescue this. In 2D, in 3D colony formation, at the level of metabolites, at the level of lipidomics. So this is the lipidomics profile of cells re-expressing wild-type PANK4. This is the lipidomics profile of cells expressing the phosphatase mutant. In a seahorse experiment, remember, coenzyme A is required for proper mitochondrial respiration. Expressing wild-type PANK4 impairs mitochondrial respiration. The phosphatase mutants do not impair mitochondrial respiration. Histone acetylation, the wild-type PANK4, impairs H3K14 and H3K27, the phosphatase mutant does not. It's a weak effect, but this is not the only mechanism that regulates histone acetylation. And finally, the wild-type PANK4 suppresses tumor xenograft growth. The phosphatase mutants do not. So we ultimately came up with a model in which cells stimulated with insulin activate PI3 kinase AKT. PANK4 is a suppressor of this pathway. Phosphorylation by AKT relieves this suppression, allowing flux from B5 to make CoA. We think PANK2, which we've studied a lot less, is a positive signal which initiates this mechanism. And now you make CoA. To get all the way to acetyl-CoA, you need an extra step, and you need ATP citrate lyase. Okay? ATP citrate lyase is also regulated by phosphorylation. What is that event? Guess what? It's also regulated by AKT. How do you get citrate for ATP citrate lyase to work? From glucose. How is glucose taken up from cells? Through glucose transporters that are regulated by AKT. That fuels glycolysis. How is glycolysis regulated? By AKT, both transcriptionally and post-translationally. Every step in this pathway has evolved over time to be sensitive to insulin and the regulation of PI3 kinase AKT to ultimately fuel acetyl-CoA, which is required for TCA cycle, histone acetylation, de novo lipid biosynthesis, and multiple other, and ultimately in aggregate, mechanisms that control cellular proliferation, which are altered in rapidly dividing tumor cells. Sorry I've taken up so much of your time, but I hope it was worth it. Let me finish by the most important slide, which is my lab. This is our website, my Twitter handle. This is my group. The people who did the work were Emily Erickson, who's now moved on. She did the work on the degrader, Sam Barrett and Christian, who've also moved on, and Alessandra, who did the work on the AKT synthetic lethal screen. These are all the other people in my lab whose work I unfortunately don't have time to show you. Very importantly, the folks who fund our work. And if you'll just humor me for 30 seconds, as was mentioned earlier, I think, in addition to running a lab, I also run a journal, the Journal of Biological Chemistry. I'm the editor-in-chief, and I always show this as a plug, but it's an important one, especially for the trainees, because I am a firm believer in publishing for academic society journals, okay? And the JBC is a poster child for this. It is run by scientists like me. Our associate editors are all working scientists. Our reviewers are all working scientists. We are not a publisher, all right? We are full open access. The time to first decision is about 2.5 weeks. When was the last time you submitted a paper to a journal and you got a decision in 2.5 weeks? Never, right? Its value, our open access fee is $2,300 compared to some of the other flashier journals. And everything, all that money, those $2,300, they don't go into the pocket of a publisher. They go to the society, ASBMB, and they use that for outreach, for running meetings, for providing grants, all right, for advocacy, for public awareness. Okay, it goes back to science. Okay, and we have lots of other programs. So if you're interested, come talk to me, send me an email. I'll be happy to tell you all about why it's so cool to publish in the JBC. Thank you for your attention. I'm sorry I took up so much of your time.